So good morning, everyone, or good afternoon, or good evening, depending on where you are. I am Emmanuelle Pauliac-Vaujour, the scientific attaché at the French Consulate in San Francisco. I'm very excited to be moderating this QBI TV panel discussion today, featuring Jason Sello, professor at UCSF, and Iuliana Ene, assistant professor at Institut Pasteur in France. Through their respective institutes, Jason and Iuliana share a scientific collaboration which has led to the recent publication of their work focused on how small molecules restore azole activity against drug-tolerant and drug-resistant Candida isolates. So we are thrilled to welcome our audience today. I encourage you to ask your questions through the chat, and we will have an opportunity to take them later on in the conversation at the end. But before we dive into the discussion about this remarkable research, just a few words about this transatlantic collaboration, which was initiated between Institut Pasteur and UCSF QBI several years ago, and truly swung into full gear in the wake of the pandemic. So the French Embassy has played a humble role in facilitating this research at a time where cross-boundary collaborations had become quite difficult. Which is why we have been very proud, particularly happy to see these joint efforts become a long-term and transdisciplinary, high-achieving collaboration. And without any doubt, today's discussion will perfectly illustrate how the collaborative foundation that has been built between UCSF QBI and Institut Pasteur serves as a catapult for breakthroughs in science and helps scientists maintain productive partnerships beyond borders. So now let's hear more about all this from the experts. So Jason and Iuliana, it's great to be here with you today. I'm going to start by asking you to introduce yourselves. Maybe Iuliana, if you want to start.
Sure. Thanks, Manuel, for moderating this panel, and thanks QBI for the opportunity to speak here. So I'm a junior group leader in the Department of Mycology at Institut Pasteur, and our group is focused on understanding how fungal pathogens, and in particular Candida species, respond and adapt to antifungal stress and antifungal drugs. But in addition to this, we're also interested in leveraging this knowledge to develop new antifungal drugs, and it's in this direction that we found synergy with Jason towards developing some new molecules against these pathogens.
Jason.
Thanks, Emmanuel. It's really a great pleasure to be here. I'm Jason Sello. I'm a professor in the Department of Pharmaceutical Chemistry at UCSF. I guess would call myself a chemical biologist. I'm very much interested in how small molecules can be used as therapeutics. And I have broad-ranging interests from the discovery of molecules that are useful for treating infections, all the way to the discovery of molecules that are useful for treating neurological disorders. So I'm really, in the sort of the spirit of Pasteur, very much interested in these sort of intersections of chemistry and biology, and I think and medicine. And I think what Jules and I have been doing together for the past couple of years has been really reflective of that.
Thank you very much. So I'm going to start with a first question. Your recent published work studies fungal infections and specifically Candida. So could you tell us more about Candida infection and its current treatment?
Sure, I can chip in. So I'll backtrack a bit to say that most Candida species exist as normal residents on our skin, on our gastrointestinal tract. So they're really part of our normal microbiome, and they don't cause any issues to our health. But when the immune system is weakened, when we take antibiotics, they can start causing infections. So these range from a wide variety of infections, from mucosal, superficial infections to very serious infections that can actually be life-threatening. So for example, bloodstream or deep-seated infections. And Candida species are actually the #1 causative agents for nosocomial fungal infectious agents in hospitals. So they're a big issue in hospitals. They cause over 1.5 billion fungal infections, and these infections lead to about 1 million deaths each year. In addition to this, we have very few antifungal drugs. So we only have about 4 classes of drugs available. Many of these are toxic to the human host, so we need to develop better drugs. And we actually have resistance reported against all of these different classes of antifungal drugs. And in addition to this, we also have different Candida species that are actually intrinsically resistant to some of these drugs. So a prominent example is actually Candida auris, which is a novel, recently reported species. It was first reported in 2009, and it's intrinsically resistant to several classes of drugs. So it's really hard to treat these infections, as I've said, because we have very few drugs available, and fungi are particularly apt at adapting and developing resistance to these molecules. So we really need better and novel therapeutic strategies against them.
Thank you very much. Jason, did you want to add something?
Yeah, I mean, everything that Jules says is absolutely on point. This is a major problem. How do we deal with these drug-resistant fungi? Is a real problem, and so we've actively been thinking about ways in which to do that. So, you can develop new antifungals, but that turns out to be quite challenging because humans and fungi actually share a common ancestor, and so a lot of the biology of the fungi has close alignment with the biology of humans. And so it's difficult to interfere specifically in a fungal process without actually interfering with the analogous process in humans. And so it's really like threading a needle to try to develop new antifungal drugs. And so I think that's kind of led us to this strategy of saying, well, maybe instead of trying to discover new drugs, maybe if we can figure out ways to deal directly with the resistance problem. If we can find molecules, for example, that can restore drug susceptibility of existing drugs, then that could be a powerful approach. So that was what Jules and I were thinking about as we started this project.
And this type of approach is actually quite interesting because it has the potential to extend the life of our current drugs, and by preventing resistance developing against these drugs, we can use them for longer in the clinic. So this combinatorial strategy is actually very attractive, and it's used against a lot of other diseases beyond infectious diseases.
Yeah, the idea of cocktails, right? Combining multiple drugs to sort of limit or constrain or circumvent resistance is quite common. We see this with HIV, for example, right? We know that the prevention of AIDS often involves cocktails of drugs that have different mechanisms and modes of action. So the work is sort of akin to that.
Right, thank you. So did you want to go into more details about the findings really that are published in this paper?
Yeah, maybe I'll just sort of start and just kick off what, sort of how we had the process of discovering these molecules. So again, our strategy was to try to figure out if we could discover molecules that could restore drug susceptibility to drug-resistant strains of Candida. And what we really were thinking about was resistance to azoles, because that's a major class of antifungal drugs that turn out to be quite useful in treating those infections. So what we envisioned was doing a screen where we could actually take a drug-resistant strain of Candida and look for molecules in a collection, screen them, looking for molecules that could restore drug susceptibility. And so we screened through, hundreds of compounds, and we're very sort of fortunate, as Pasteur says, chance favors a prepared mind. I guess our minds were prepared, and so chance was in our favor, and we were fortunate to discover some molecules that were able to restore azole susceptibility to resistant strains of Candida albicans. And what was more exciting about that was that the compounds also could potentiate or increase the potency of the azole drugs against azole susceptible strains of Candida. So that was exciting. But then there were a number of sort of findings that really were sort of pushed, pioneered by Jules to sort of explore this. So, for example, we found that the compounds themselves, although they could make the azoles more potent, the molecules did not have any measurable antifungal activity themselves, these potentiators. So we were excited about that. And then we went on to show that those compounds had no toxicity to human cells, which is very important in terms of thinking about sort of the next steps. So after having shown that, I think some very beautiful microscopy experiments were actually done within by Jules's team, which is able to show that the molecules, although they were not toxic to Candida, they caused aberrations or defects in filamentation. So these Candida is able to adopt sort of a filamentous morphology, and that filamentous morphology is associated with pathogenicity. And so the fact that these molecules are able to suppress filamentation, this sort of virulence or pathogen-associated, morphological change, was really quite fascinating. So again, sort of looking very carefully under the microscope, that became evident. And I guess the other sort of thing that was interesting was doing some microscopy. Again, it became evident that these molecules interfere with sort of lipid homeostasis. And so there's some pretty different distributions of lipids within these Candida that had been treated with these compounds. And so we think that these molecules will be useful from a potentially from a clinical direction, but even just as a basic probe of biology. So how do we understand lipid homeostasis? So that was it. And then I guess the sort of the real major sort of discovery here was that we were able to show in a in vivo model of a fungal infection that these compounds were efficacious. So there's this Galeria larva, so a moth that can be infected with Candida, and it will actually succumb to those infections. And we were able to show that these molecules are able to promote survival of these insect larvae that had been dosed with these compounds. So this really is what gives us excitement about going forward with these compounds into mammalian models of infection. So Jules, do you?
Yeah, so I think what Jason said, it's a perfect summary of our findings. So I think it's also important to mention that we're really at the early stages of this work, and what we're doing right now is really to try to optimize these molecules to improve their pharmacodynamics, to improve their spectrum of action, and also their potential for really treating these infections. In addition to that, we are working on defining the precise mode of action, so on identifying the exact molecular target that these molecules target in order to, again, optimize them for improved efficacy. So as I said, this will be a lot of work that's planned for the future, and I think we're both quite excited about it.
Sounds really good. Thank you very much. So as we listen to you, we understand that this collaboration was really quite important in the having those results. So can you tell us more about how this collaboration came about and how it helped through your research, what gain you had through this collaboration?
Sure, yeah. So it's actually a funny story because we met while we were both at Brown University. So I was a research assistant professor and Jason was a full professor there, but we were in different departments. And it was actually my former advisor, Richard Bennett, who got us in touch and realized that there might be some synergy between us. But then soon after, the pandemic came, and then we both moved institutions, and it was quite struggling to continue collaborating in this environment, like adjusting to a new institution and also dealing with the fact that our labs were shut and we had to quickly adapt to this while maintaining our research programs going. So it's really worth mentioning that the people in our labs who are very talented chemists and biologists that actually helped push some of this research forward and kind of brought it to its fruition. But other than that, we really synergize by having expertise in different fields. So I'm a molecular mycologist, whereas Jason Sello, he's a medicinal chemist, and we've learned a lot from each other. This is my first collaboration with a medicinal chemist, and I feel like every time I talk to them, I'm learning something new. And the synergy between us is really based on this interest of developing better antimicrobials. And because there's such a high need to develop better antifungals, I think this is really has been the real motivator behind it.
Yeah, that's absolutely right. I think, as Jules was telling the story, I can't help but think about my student, Philip Alabi, who was really a driving force in putting this project together. He was the one that made the compounds, was ensuring that we really were working together, as Jules mentioned, during a very sort of challenging transition that both of us were making during the pandemic. But I was really I'd worked in bacteria for years, and I just reading the popular literature and seeing just how badly new antifungals were needed, and I thought it would be an attractive area in which to work. And there's a pretty high bar for someone shifting from bacteriology or medicinal chemistry into working with fungi. It's a completely different infrastructure. It's a non-trivial thing. And so to be able to partner with Jules, who had the expertise in being able to do everything soup to nuts, from genetic manipulation of fungi all the way to characterization of their morphology using microscopy, it just was a perfect partnership in order to be able to push this forward. So it really does take people with complementary expertise to do this. So it's been really fun. I've learned a great deal as well from Jules about mycology.
Right. Okay. As a scientist myself, I'm very happy to hear that multidisciplinary collaborations are key, and yeah, because this is so important today. And well, actually, that leads me to the next question, which is that, as you know, UCSF, QBI, and Institut Pasteur have launched this initiative to create a Center for Excellence on Emerging Infectious Diseases in the Bay Area. So this will definitely foster some multidisciplinary research, but I was wondering how this would help you, how important it is for you to go on collaborating within the frame of this center, and how feasible it is to actually collaborate across borders.
Yeah, I think to date, Jules and I have been sort of doing this, just with just individual funds that we have together. And I think there's been, we've made actually a great deal of progress considering a small amount of resources that have been put together. But being able to associate with this sort of joint institute between these sort of two august institutions really is really setting the stage for exciting stuff. So, for example, I had not seen Jules and I had multiple Zoom calls over the past couple of years, but by virtue of this connection, we were able to meet in Paris at the Pasteur and actually discuss work and plan things going forward. We're also interested in the possibility, the fact that there are funds available for exchanges. So I'm very excited about the possibility of hosting Jules here in San Francisco in September for a time for us to sort of work together, for us to have more in-depth discussions, but also for us to talk about experiments, but to plan for grant proposals as well. So we're in the process now of putting together a grant proposal that will really enable us to accelerate the work at a rate that's much, much faster than what we've done before. So to actually have financial support for our collaborative efforts, joint financial support, will really push things very quickly.
Yeah, and maybe to add to that, I think it's really exciting for us to travel and meet the other scientists at our respective institutions. So when Jason came here for the symposium in April, he was also accompanied by about 20 other scientists that I got to meet and talk to, and discovered other potential synergistic avenues and potential interest for collaborations and putting other things together. And I'm also very excited to meet the scientists at UCSF and to see what tools they're using, what questions they're interested in, and how they are working to address them. So I think it's really this idea of building bridges together, so beyond countries, institutions, and also disciplines, which I think is going to make our research a lot more productive than it currently is.
Okay, that's great to hear, actually. Hopefully some young researchers also, such as you, Iuliana, will get involved and foster this type of of exchanges. It's particularly important for the French embassy to see young researchers traveling from France to the US and US to France. So I'm pretty happy to hear you say this. Right. Unless you want to add something about your research, maybe we can take some questions from the audience, if there are any. They should appear on the screen. Right, so we have a first question, which is, how will you move from larvae to patients? And how can you study the efficacy of the antifungal drug in patients?
Yeah, that's a great question. And I think this will be a big leap in our research. So what we're doing right now, we're moving from larvae to mice, because this would be a great first model and a first demonstration of whether or not these compounds have potential in mammalian systems. And then I think there will be a lot more improvement over these compounds and a better understanding of their pharmacodynamics, their dosage, their application before moving into patients. So I think we're really at the very early stages of this direction. And first, we're excited to see what happens in mice and a variety of different models of infection, because as I've said initially, Candida can really infect humans in a lot of different ways. And we have already established several different fungal infection models that can mimic some of these infections, from oral infection to systemic to gastrointestinal, a lot of different models that can be used before transitioning to patients. Great question.
Jason, did you want to add something?
Yeah, I think, as Jules said earlier, these are the very early days in this, and there are many different indications. And so trying to figure out for which indications these molecules will be most appropriate will actually take quite a bit of work. So, so this is one of the reasons that we are excited to put together grant proposals that will give us the funding in order to do that critical exploratory work that will define the clinical utility of this. So, you know, we started this because of its clinical utility. And so we're really committed to seeing this borne out and, you know, in that way to sort of really establish what's, you know, what the use of these compounds will be.
Thank you very much. So we've talked quite a lot already about, you know, what the future may hold for your collaboration. Maybe I'll just ask one final question, like what would be your dream breakthrough? What keeps you awake at night?
Yeah, I can start. I mean, I would really like to have something that works against a variety of different species, so something as broad spectrum as maybe azoles. But we're, of course, too far from that right now. So azoles are really nice because they work on a variety of different genera of fungi. So anything from Candida, Aspergillus, Cryptococcus. And this is really what we're targeting with, or what we're hoping for when thinking of like what's an ideal antifungal. So of course you want this to be broad spectrum, but you also want this molecule to have low toxicity to humans, to be able to be administered orally rather than require IV administration, all sorts of practical things that actually might break a deal between making or not making a new drug.
Yeah, absolutely. We've done this. We've shown this work success with azoles. I keep telling Jules, it looks like we have maybe a little bit of glimmers of hope for something that's active against Candida auris, which is really making an increasingly big impact, particularly in hospital settings with the elderly and immunocompromised in particular. So I'm excited about that. But in principle, we can apply this same strategy to the other major categories of antifungals, and maybe have a suite of molecules that can be used to potentiate the different classes of drugs. So so those are the kinds of things that we've talked about, sort of really expanding this out.
Great. And well, actually, we've got a question popping up, which comes quite nicely into the discussion. How do you see this work moving forward? What are the next steps that you're envisioning?
Yeah, so the first step, I think it will be absolutely important to identify the molecular target of these drugs, and also to optimize them for increased efficacy on Candida albicans. But beyond this, to expand our spectrum of action to other Candida species. So we're particularly interested, as Jason said, on Candida auris, but also on species such as Candida glabrata, which is also intrinsically resistant to azole drugs. So I think the broader the spectrum, the better the molecule. So in addition to this, we'll need to define the modes of formulation, delivery, indication for such molecules, and really demonstrate that they have in vivo applicability. So I think these are the immediate next steps, as well as for us to be able to obtain some research support to carry on some of this work.
Thank you very much. Jason, did you want to add one last thing?
Stay tuned.
Absolutely. Absolutely. There will be much more to see in the future, I'm sure. So I think we don't have any more questions. So we will probably wrap up our discussions here for today. Thank you, Jason and Iuliana, for your time. Thank you as well to everyone who tuned it. You can obviously see the video again later on and react to it. Feel free to leave a comment. It was my pleasure to moderate this interesting conversation. I look forward to the evolution of this fruitful Franco-American collaboration, which we are particularly proud of. Well done. And if you're new to this channel, please subscribe to QBI TV for future events. Thank you, everyone. Have a good day, evening, night, whatever.
Thanks for having us.
Thanks for having us, Emmanuel. Yeah.
See you next time.
Okay.
Bye-bye.
Bye.